Hi guys, so continuing chapter 13, part 2. Now we are talking about multiplication of bacteriophages. So these are viruses that multiply inside bacteria. They go through two phases or through two cycles. The lytic cycle, where the phage lyses the bacterial cell when it's multiplying and when it's exiting the cell, and the host cell dies. And the lysogenic cycle, where the phage DNA is incorporated into the host DNA. And that can lead to three important events. One is not mentioned here, but we're going to talk about phage conversion and specialized transduction. This lysogenic cycle can also cause immunity, which means the bacteria that incorporates that viral DNA gets immune to getting infected by the same virus or by other types of viruses. So the bacteria no longer gets infected by viruses because it's in the lysogenic cycle. Okay, what is that? Let's talk about each one of them. Okay, so I'm gonna show you the picture because I'm super visual and I hope you are too, and I think that's gonna make much more sense. Let me zoom this. Let's look at this. Let's look at what's happening here. You have a bacteriophage here, correct? It is going to inject the DNA, which is this purple genetic material, into this bacterial cell. This is the bacterial cell, right? Remember, purple here is going to be the bacterial DNA, okay? Actually, they make both DNA look purple. So yeah, in this case, let's just not think about the color. The phage is injecting the genetic material into the bacterium, right? This is the bacterial DNA, okay? The phage, once the genetic material is in, what is carried inside this capsomere? DNA and DNA polymerase. So what is the virus going to do? Use the machinery for replication of DNA in the bacterium to duplicate the DNA first. Second, to transcribe RNA for proteins that make all these other parts of the virus. Make the proteins through the ribosomes of the bacteria for all these parts. And in the third phase, assemble them. So first phase, duplication of DNA. Second phase, make an RNA and make the proteins, right? Make the proteins that are going to be part of the newly formed viruses. And then third phase or fourth phase, everybody gets assembled, and we call this maturation. And release or exit is when the bacterial cell is lysed, it ruptures open, the bacterial cell dies, and once you had one virus, now you have 1, 2, 3, 4, 5, 6. Okay, so one becomes many, and they lyse the cell and they exit the cell. What do they do next? Infect another bacterium. And that's the whole process. So if I make this smaller, you're gonna see here just in detail how the tail, the capsid, and all the parts are formed, okay, and put together during assembly or maturation, okay? So this is how a bacteriophage completes the lytic cycle inside a bacterial cell. Now we're going to talk about lysogeny. What is this? It's when the phage becomes latent inside the cell, bacterial cell. So the phage DNA incorporates into the host cell DNA. It inserts the phage DNA, which is known as a prophage. Let me show you this. So this was the lytic cycle, right, that we just talked about. Now I'm going to show you this part of the cycle here. This is the lysogenic cycle, okay? So if you look at this, you're going to see see if I can move this for you. No, I can't. You're gonna see that there is the purple DNA is the bacterial DNA, and in this case here, the pink DNA is the viral DNA, right? So the viral DNA, the phage DNA, incorporates into the bacterial DNA. And what happens, so they were separate, and then if you look at this part, they're incorporated. So you have a prophage, which is the viral DNA incorporated into the bacterial DNA. Every time the bacteria multiplies, it multiplies the prophage. How smart are viruses? They don't need to do anything. The bacteria is doing all the work for them, and they're just latent and sitting in that circular DNA. So is this the virus? It is, because the virus is the DNA. From that DNA, one day that virus can undergo like all the cycle, and there are some signals in the cell that determine if this cell is going to continue in this cycle or if it's going to undergo lytic cycle. If it goes to the lytic cycle, what happens is that the DNA from the virus excises the circular DNA of the bacterium, makes new virions. Virion is, remember, each particle. And those virions can be released and they kill the cell, the bacterial cell. So between lytic and lysogenic, there are signals that are encoded by genes in the bacterial cell that are going to allow the prophage to be there sitting for long periods of time or not, depending on the virus, depending on the bacterium, okay? And usually you have bacteriophages that are very specific for some bacteria species, okay? They usually infect a specific species of bacteria and not the other ones. With this, what else I want to tell you? That sometimes during the lysogenic phase, the bacteria undergoes what we call phage conversion. Phage conversion here is, right here, what? The bacterium changes and acquires new properties because of that provirus. Because of that prophage. Provirus is how we call it in animal cells. And then what's gonna happen? These are examples here. For example, the Vibrio cholerae, which is, as you know, the bacterium that causes cholera, if it receives a CTX phage that encodes for cholera toxin, so this came from where? From the virus, right? From the phage. If the virus gives the gene for cholera toxin to a Vibrio cholerae, then that Vibrio cholerae is going to be super pathogenic because the cholera toxin is what causes that watery diarrhea. And then phenotype, the bacterium causes cholera. So there are other examples of bacteria that are not pathogenic, like E. coli, when it acquires by lysogenesis Shiga-like toxin, is going to cause hemorrhagic diarrhea. Otherwise, it's not. So by lysogenic conversion or phase conversion, bacteria can become virulent and pathogenic. Does it make sense? Another process that is also can be a consequence of the lysogenic cycle is the specialized transduction. In this case, it is similar to the phage conversion, where the bacteria is going to acquire a new feature. But here, the DNA that the virus is transmitting or injecting in this new bacteria comes from another bacteria. It doesn't belong to the virus. So look at this. You have this bacterial cell, and this bacterial cell right here has a prophage, right? So once that prophage excises and gets out of this bacterial number 1, it brings a tiny, tiny piece of this bacterial DNA with it. And then when it injects the material into this bacteria number 2, a little tiny piece, you see this purple piece, is from bacterial number 1. So when does this usually happen in bacteria? It could be that you have specialized transduction, transferring like things that can be related to different functions in the cell. For example, the example that the book gives is the galactose metabolism. There is a gene called GAL that produces enzymes for galactose fermentation. This bacteria can do galactose fermentation. The second bacteria cannot until it undergoes specialized transduction, and then it can, because it received a little tiny piece of bacterial gene from bacteria 1 that codes for the GAL gene. Who brought it to the bacteria number 2? The phage. Does it make sense? You understand the difference between this and the phage conversion, where the gift, whatever gene that the virus, the bacteriophage is transferring to the bacteria, comes from the virus, not from another bacterium. Okay? So we're going to see different diseases where this happens, like diphtheria is another disease where you have phage conversion and the bacteria becomes toxic, becomes pathogenic because of that toxin. Okay? Next video, we'll talk about animal viral multiplication. So I'll see you in a minute.